Welcome to the next episode of Engaging Human Science, brought to you by Animal Free Research UK. Animal Research UK is the leading charity working towards creating a world where human diseases are cured faster and without animal suffering. My name is Geoff Pilkington. I'm Emeritus Professor of Cellular and Molecular Neuro-Oncology, based at the University of Portsmouth, but also at King's College London and the University of Cardiff. And I've sort of just about completed a 50-year career in neuro-oncology. So I'm a career neuroscientist in that aspect. I'm interested in the use of repurposed and reformulated drugs, particularly for the treatment of cancer and particularly brain tumors. And I've been working in an animal-free environment for probably 30 plus years of my career, developing 3D all-human models for blood-brain barrier, for example, and preclinical testing. So that's me, but more importantly, it's a pleasure to introduce Professor Amanda Ellison from the University of Durham. Professor Ellison is Professor of Neuroscience in the Psychology Department at Durham, and she's also Director of the Wolfson Research Institute of Health and Wellbeing. She has interests including the neuroscience basis of vision, neuronal disruption in migraine headache, visuomotor systems, sensor-motor cortical interfacing, and a lot of other things. I'd like to explore these other things in more detail as we progress through this interview. So, welcome, Amanda. It's nice to see you again. We have met before. We've met before on a number of occasions, but not at the distance we are at the moment, because Amanda's up in, I think, Middlesbrough, overlooking the North Sea, and I'm in the Isle of Wight, overlooking the Solent and the English Channel. So we're about as far apart in England as we conceivably could be. So anyway, what I'd like to start off by talking about is this is fascinating for me, that you're working in a psychology department. I worked in the Institute of Psychiatry for over 23 years, something like that. So I know what it's like to have different hats. But you seem to be multifaceted because you have various different roles. And I wondered how you managed to keep all this together with your different interests from a sort of an administrative as well as a scientific career point. Can you give us a clue as to how that all holds together?
Yeah, it's interesting. I'll tell you one thing, it's never the same bite twice. So no 2 days are the same, that's for sure. I think that the words that you were looking for when you were describing my varied scientific interests were that I am scientifically promiscuous, which I am. But actually, it's born out of curiosity, really. And it's also born out of one of those insatiable needs to understand every facet of a problem. And of course, that opens up all sorts of other doors. It's very Alice in Wonderland. And then that opens up lots of other doors. So it's unless we understand all of the aspects of a problem, we can't actually address it. So ultimately, whilst I'm interested in understanding how something works, it's always with the general aim to fix a particular problem. But by understanding how different parts of the brain talk to each other, for example, then that means that my finger gets to be in lots and lots of different pies, from addiction, through hallucination, through pain, all of those varied things that seem to be completely different, but actually they're not.
So I mean, that's intriguing. And I found that I've just been reading your book, and forgive me, I was reading a book by Johnny Wilkinson, the rugby player. Johnny Wilkinson and I went to the same school, albeit 28 years apart. And so I was interested in reading what he'd done. So I put that down and came over to this, which is completely different, but actually
an excellent company.
You know, very, very similar in my interest. However, I mean, there were certain things that came out of this which I can completely relate to. So, you know, in particular, your description of your stressful, packed, almost impossible typical day, you know, the scenario there, and I can relate to that as a neuroscience professor myself. That's what it's like for many of us. And I've had 2 years now to contemplate retirement and so on. and I'm still got my L plates up, because the only difference really is that I'm not being paid for it. But once you're driven, and you obviously feel that drive in your career, and what I'd like to explore a little bit about is, you know, what brought on that that quest for achievement and knowledge and so on. So what stimulated your interest in neuroscience from day one?
Well, you know, I trained in physiology. So I have a natural science degree. So, you know, when you do one of those, a 4-year degree at Trinity College in Dublin, so, you know, lots of time to learn about different things. And then in the end, I moderated to physiology, which meant I got to, in the 3rd year, do lots and lots of things and see how they relate to physiology. And then in the 4th year, it's all about dissertation and actually solving a problem. My dissertation was in sports physiology. So what I actually did was I got in semi-professional cyclists and then stuck them in a sauna for an hour and then thermally dehydrated them and then had a look to see how that affected their performance. Now, this was before we knew how important drinking water on the pitch was for footballers. And, you know, there were like World Cups in really hot places, and people couldn't quite understand why their players weren't performing. And it was because they were dehydrated before they even started. So it fed into that kind of narrative. And I was hooked, because up until then, I kind of didn't feel like I had a question of my own. And then all of a sudden I had this question that I asked and answered. In those days, even in an undergraduate, we had a viva for our dissertation. So I had an interview and I had an external say to me, it's too good. It's a linear effect. The more dehydrated you are, the worse you are, the less you can work. And I'm like, yeah, I know, it's amazing, isn't it? And he was like, no, you made it up. You know, so like, it's like, what? So I knew that actually, even from then, if you were going to be a scientist, you had to be resilient. You had to fight your corner with evidence. And I think the evidence is the key. I didn't stay in sports physiology because you had to be a medical doctor at that point to go into a postgraduate in sports physiology, which doesn't exist now, but it did then. And I did want to do something about the brain. As a physiologist, I was very interested in how all of the different organs work together. You know, we had 10 lectures on the kidney and 10 lectures on the heart, but my mind was saying, yes, but how do they interact? And that's the really interesting thing. Now, the brain, as you know, all those years ago, I mean, we're talking, when did I do my undergrad? I started my undergrad in 1991, so like yonks ago. And in those days, neuroscience didn't really exist as its own discipline, certainly didn't in Ireland. You had physiology, you had neurophysiology, and you had psychology. And psychology was behavior, and physiology was mashing up brains in a petri dish or in a test tube, adding reagents, seeing what happened, you know. And I wasn't interested in that. For one reason, I couldn't see it. I need to be able to visualize what it is that I'm doing. And also, for the second reason was, I didn't think that we could ask or answer any questions that warranted the sacrifice of an animal's life. I just didn't want to go down that road. So, but I did want to do whole brain physiology. I wanted to know what different parts did, how they talk to each other. Made a lateral move into the psychology department where, you know, they were just beginning to think about things like that with respect to the brain. Took lots of stick for being a physiologist in a psychology department from both directions. But, you know, I followed the questions that I wanted to answer, and eventually I went some way to answering them. I don't want to answer all of my questions yet, because then I don't have a reason for being. But luckily, I keep finding new questions, and questions beget questions.
I don't think there's ever an end to it. I mean, there's a big world to explore out there. I mean, I your point about the being able to see things, and unfortunately, the way things are going in science, we've gone through this molecular era now, where we can't see things. We have to take the assays as read and so on. So I find that very difficult as somebody who came out of a morphological training originally in pathology. So seeing it is quite important, and unfortunately that's happening less and less. But looking at the situation as far as neurophysiology is concerned, I mean, I worked in Department of Clinical Neurology at Queen Square, the Institute, National Hospital for Nervous Diseases as it was then. And virtually 100% of the research was carried out on living animals or people, mostly on living animals. And that's changed out of all recognition. Can you give us your thoughts on how that change has happened and what you can do to change that towards a more human approach?
Yeah, I think there's two ways of looking at it. One is that we now have alternatives to that. So, you know, I came up through the genesis of transcranial magnetic stimulation, which sends very brief magnetic pulses into the brain. Switching it on means that it actually acts as if it's not there. So you can ask really discrete questions about the brain. We have, you know, we've always had neuropsychology. We've always had that with humans. You can wait until somebody has a stroke, or they put their head through a windscreen, or they have some other kind of closed head injury. And, you know, in the early days we had, and by early days, I mean early days of my career, we had things like positron emission tomography. fMRI was starting to come online. Certainly MRI was there. They gave you really nice pictures of the brain. PET less so, because it was a bit blurry, and you also had to inject people with pretty much radioactivity in order to see the pictures that you wanted. I did a PET experiment when I was at Oxford, and you know, you couldn't include any women in that, because there's always the chance that they could be pregnant, and this would hurt the fetus. You had to use men over the age of 40, because you couldn't trust men below the age of 40 to be able to make the decision that injecting themselves with something that's radioactive would actually hurt them or make them sterile. So either men over the age of 40 don't want to reproduce anymore, or the men under the age of 40 weren't capable of making the decision. I don't know which that is. But you know, you were quite restricted in the people that you could use for these experiments. So, you know, you're a scientist, and any scientist listening or watching will know that actually, you know, you don't want to constrain your sample that way if you want to ask questions about humanity. So, you know, we had these other techniques, like animal techniques, animal lesion techniques, or things like that, or listening to the electrical activity that was going on in a behaving animal. But, you know, as our knowledge of other ways of doing things with the human improved, so things like transcranial magnetic stimulation, which does exactly the same thing as what a lesion technique would do. It's a lot more discreet than neuropsychology, where you've got a big lesion that's happened unnaturally to the human, but actually is something that the human is constantly trying to overcome. So they've got all sorts of rewiring and compensatory mechanisms going on. So they're not a great way of working out what does what. So that's why neuroscience turned to animals, because if you lesion here and work out it does that, then that's great, you know. But TMS will help you do that as well. And so that then, over the course of my career, became a very credible alternative. And then putting that together with what it is that you find in fMRI, because that's come on in leaps and bounds, and sometimes you put the two together. And I think that this way of working and thinking in a more interdisciplinary way about how it is we address our problems, it's not enough just to address them maybe with one technique. You can answer the question more fully and more holistically with two. But then in that way, it's much more relevant to the human, which is another driver, and has no recourse to the animal.
So looking at the imaging techniques which are available at the moment, and the evolution that's been fairly rapid over the years, if we're trying to get that important transition between diagnosis based on histology, based on a range of different molecular markers and so on and so forth, trying to recapitulate that system into the living individual and trying to see what's going on. So we get little snaps of what's going on in any given pathology. How close do you think we are to making that one big continuum, so that we can then diagnose and perhaps treat, you know, getting the appropriate agent into the appropriate parts of the brain or cells of the brain and so on? How much of a challenge do you think that will be, and do you think we're going to reach that in the foreseeable future?
I do hope so. I'm working on a project at the moment to do with aneurysms, and one of the biggest problems that we have with aneurysms is spotting the aneurysm from these snapshots, as you say. And an aneurysm is like it's a bulb in a blood vessel, and it destabilizes the flow of blood through that blood vessel, and the chances are that that little bulb will burst, and it's called a rupture, and it causes, in effect, a stroke, a bleed on the brain. And so, you know, it's really difficult. And the other problem that you haven't mentioned, but I think is implicit in what you were saying, is that really when we do these things, and when we write these papers and when we submit them to journals, it's almost as if it's moved the field forward a little bit, which is what our papers should do, it generalizes to everybody. And we're not talking about everybody, we're talking about the people that we tested, right? And so that means that inherently, and from the very beginning, we're only actually speaking for some of the people some of the time. So what we really need to do is to start looking at the whole plethora of questions that we ask about all the different conditions that we investigate from a very individual point of view, from that patient-centered perspective. And only then, by putting together all of that data, so what we do, like with the aneurysm work, we've got a lot of background data, psychosocial data, you know, things like, you know, what's your economic status? What's your educational status? How many people do you have at home? Do you have a family? Have you had kids? Do you have parents? Do you have dependents? Do you have a dog? Do you believe in God? All of these things can actually affect how it is that our brain works. And some of those may be active or not at that snapshot that you take, that you're looking at. And until you understand the variety of the personal experience, I don't think we're going to get anywhere close to be able to understand what we're actually looking at. So we need to be able to attach all of that data into what it is that we're looking at. And once we do that, I think we'll see a big change, because I think then machine learning and everything is going to take off, and we're going to be able to diagnose these things in a much more automated way, but in a much more nuanced way as well.
So perhaps this holistic approach to medicine is particularly pertinent within the neurosciences, it being the control center over everything else. So yeah, maybe that's the first sort of wall to attack.
Yeah,
but it's an interesting perspective, and I think the technology has been in advance of us for a long time, but gradually the biology is on catch up. So should we just go back to your book, because I was fascinated to read that. I have to say I've only read half so far. I will complete it, but I'm a very slow reader and I've only got halfway through. However, the thing that struck me was not only was it written in a sort of a scientific way, but also in a very, very humanitarian or humanistic sort of way, that it was far-reaching in almost like a novel, or part of your life was coming out there, as well as the scientific information. So it was very eminently readable. But the thing that struck me most was that the diversity. You say, well, I'm a neuroscientist, that's fine, but then you go into the neurobiology, you go into the neuroanatomy, the histology, the cytology, the psychology, the pharmacology, the immunology, the physiology. So you are bringing in so many diverse sciences into that. And actually, and that's very rare that you see in a textbook or a book which is addressing a particular disease or series of diseases, as indeed this does. It's a very different take on a scientific, almost a scientific novel in a way, but it's actually not fiction. It's scientific fact, and you bring in all the characters and the personalities who you've met through the years and learned from and with and so on. So it's fascinating. So what gave you
lovely
making that documentation? How did you come about that concept, or did it just happen?
Well, it just happened. I'd written a book before, and it was called Getting Your Head Around the Brain. And it was, again, very, very much in that whole, right. The problem that I had with some books that I'd read on holiday that year, I think it was 2010, and I'd read some books on holiday, which is, you know, as you know, that's sacred time. That's the only time that you get to read the really interesting stuff to expand your mind outside of the kinds of scientific stuff that you would ordinarily read. And I was really let down by some scientific writers, because I felt like, do you know what, I've learned nothing from that. I've learned, and it wasn't because of what I already know, which is relatively little in what I was actually reading, but it's more about just describing stuff is not enough, right? And it annoys me, because you can describe a patient's experience and leave it there, and lots of books do, and they do very well. Whereas for me, what I want to do is describe an experience and then say, and here's why that happens, you know, and here are all the things that, this is how it works, you know. So this is why I wrote Getting Your Head Around the Brain, because what I wanted to do is try to explain what's happening in this bit of real estate that brings about all of your different behaviors. So I've done that, and that was great, and I quite enjoyed that. And then I was asked if I ever wanted to do it again. Now, the first book that I wrote was a hybrid textbook, so it was for students primarily, and it's you know, a small hang on, I'm going to find it for you. It's just a small volume. Here it is. Okay, and it's only small. Look. And so it's kind of thing that you could read on the loo, right? Fine, but it's not a big tome. It's not a big textbook, but it kind of brings people into the topic by engagement. That's the key, I think, because to me, as scientists, we have to engage people with what it is that we're doing, and if we're not doing that, that's actually a shame on our house as opposed to their house, the person who's listening to us. So it's really, really important. But when it came to this book, I was trying to think of something that would allow me to talk about the experience of what it is to be human, and how it is we are made that way, and how it is everything works together to bring about an experience of being human. And one human experience that many of us have experienced is headache. And so headache, by looking at it from that angle, I could actually unlock all of these different aspects, because we need to understand the genetics or cytology or whatever it might be. I tend not to use those words, but it's just more about saying, right, what do I need to explain this? It's a story. And as you quite rightly said, Geoff, these are all characters on the stage. In fact, don't get me started talking about the action potential, because that's exactly the terms in which I talk about it. These are all the characters, and this is the stage. And now here's what these characters do to each other. And it's like Macbeth by the time I'm finished with it. But you know, it's like, this is how we engage people. And this is how it makes it real for people, personally.
I think that's very true. In fact, forgive me, I missed out the genetics, and I certainly missed out the epigenetics from the list of areas that you addressed on the way through there, which is pertinent. I think probably at this stage we should, anybody who's listening or watching out there, we should say this is the book we're talking about. It's called Splitting. Amanda Ellison is the author. It's the subtitle is The Inside Story on Headaches, and it's published by Green Tree. I noticed from one of the other podcasts or interviews you did, which was a transatlantic one, that the interviewer was struggling to find the publisher. So it's Green Tree.
It's Green Tree, and they're a division of Bloomsbury.
Yes, I did my work on that bit as well. Anyway, that's the book. It's a fascinating read, and I've only read halfway through as well, so lots more to read. I'd like to divert a little bit now to some of the things that you brought up there, and perhaps just talk about some elements of the nervous system where I have some experience coming from a completely different stance from you, looking at brain tumors and so on and so forth. But one of the things that came up in this book quite often was the fifth cranial nerve, the trigeminal nerve.
Mm-hmm.
Which feeds into the face and so on. Way back, I did some studies on animals. The first part of my career, I suppose the first, 10 years of my 50-year career was spent there. We couldn't do anything else other than animal work. And we did all sorts of things like diphtheria toxin lesions for demyelination in animals and so on. But with the early work on tumors that I did, we were creating tumors in animals, in rats and in mice. And we found that the tumors of the cranial nerves, these things which are generally called schwannomas or acoustic neuromas if it's the acoustic nerve, but those in man are almost invariably benign in nature. They can turn nasty and so on, but they're histologically benign tumors. And yet the only ones that we could find in these chemically induced tumors, which were producing gliomas, brain tumors. The tumors were almost, the cranial nerve tumors were almost invariably in the trigeminal nerve, and they were malignant in nature. Completely different tumors, trigeminal nerve tumors, but malignant. So it begs the question of, that's a very important nerve in terms of pain and your studies on headaches. What is it that's special about that nerve that could render any biological stimulus different? Are there differences in the anatomy and the physiology and in the functioning of these different cranial nerve systems? Do you have any thoughts on that?
Yeah, I don't think in function, certainly. I think all nerves communicate in much the same way. They all need the same nutrients, all of that kind of thing. I think that the ultimate function of what it does and what it serves to do is different. So the trigeminal nerve, in effect, protects us, because that's what pain is for. It protects us. If you've been thumped in the face and your trigeminal nerve is sending off signals to your somatosensory cortex going, I think you've been thumped in the face, that's where you move away really, really quickly, because what it's protecting is your brain. It's protecting this head area, which is so important for how we interact with the world. It's protecting our eyes, it's protecting everything in this area. And if you think that, you know, with the exception of the sense, somatosensation, I'm cutting in my head because there's lots and lots and lots of different views as to how many senses we have. But if we think about the usual suspects, so we've got vision, we've got hearing, we've got olfaction, we've got taste, and we have somatosensation. And within that, we lump hapses, and we lump proprioception, and maybe interoception there as well. So with the exception of somatosensation, which governs the biggest organ in our body, which is our skin, we have the other four senses represented in this area here, right? And we've got to protect it, okay? So the trigeminal nerve is incredibly important. Why does it become malignant? Well, you'll know better than me, but I think that actually if we look at it from a neurobiology, neuroscientific, neurobehavioral axis, then you can start to think about, okay, what does the trigeminal nerve do? It indicates danger, right? So it's going to react in a particular way if there is danger around, and that can be chemical danger. So we know that that the trigeminal nerve is very sensitive to potassium, for example. Those kinds of just very, very simple ions that are around in the system.
So these tumors were being induced by chemical neurotoxins, potent neurotoxins. So that would fit in.
Yeah, exactly. And there are lots of chemical changes that happen with pain. So when we're in pain, it's a two-way street, right? So trigeminal nerve may be activated by chemicals, yes. It can be activated by cold stimuli, yes. But it can also then detect vasodilation, for example, and it sends signals to other areas in the brain, which then, hold on now, there's pain there. I'm going to have to send an immune response to that. And that immune response in turn activates the trigeminal nerve. So it seems to be on this almost hair trigger of a vicious circle. And so it's because it's constantly active and reactive. That might be one window into why it is that those tumors would become malignant as opposed to just being benign.
You see, this is fascinating because what we're looking at is animal models here, which aren't telling us the same about humans. And so there are a number of examples of that within the nervous system and within many other parts of the body. So another one of those is another one of the areas of the brain which you've concentrated on quite heavily, which is the hypothalamus. Ooh. And my observation on a tumor called a pilocytic, a hair-like astrocytoma. So these are the astrocytes, generally stellate, generally star-like cells, but some of them are like hairs, so they're long, thin cells. And generally those are found in children, and they're found in the cerebellum. But occasionally they can find their way into the hypothalamus. Now they're almost exclusively low-grade benign tumors when they're in the cerebellum, but when they get into the hypothalamus, whoosh, they take off. they're a completely different beast. The microenvironment being different in those areas. So again, there's the same sort of situation, same question, if you like. What's so special about the hypothalamus that it can change the biological behavior of what's an indolent sort of disease normally?
That's a really good example, Geoff, because both of those areas have inputs and outputs, and the cerebellum takes its input from mainly motor, but other regions as well. But it's so synonymous with it. It will correct our movements, right? So you throw a dart at the dartboard and you miss the dartboard entirely. It's your cerebellum that's thinking, okay, what do I have to do to correct that movement so that I can hit the dartboard and not my husband or wife or dog or whoever else might be there. So yes, so it's very important in feedback mechanisms, right? And that's a really interesting thing, because so is the hypothalamus. The hypothalamus controls our homeostasis. It controls through feedback how many hormones we have, and they can be anything to do with satiety, thirst, sex, all of the hormones, anything that keeps us ticking that we're not really aware of. That's what the hypothalamus is doing. But the difference between that and the cerebellum is that it's got so many more links into body systems than the cerebellum does, and so many more links into so many different feedback systems. So it's the endocrine system, but it's also the sympathetic nervous system, and it's into everything. It's why it's my absolute favorite part of the brain, but that's why it's so dangerous if something goes wrong there. Cluster headache is an example of that. But we need to understand not just about how the neurons are talking to each other, but also what neurochemistry is going on there. So what neurotransmitters are going on there, because the way people behave and the way people can behave abnormally can be through a problem with their neurons, and that can happen with the tumor, and that neurons just aren't working or they're impeded and they can't work, but also through neurotransmitters, that the differing concentrations of neurotransmitters will affect how people behave. And you need to know which it is, because oftentimes for people who aren't scientists, they'll think, I have brain damage because I can't do X, Y, and Z. But actually, it could be just because, and I say just because, I don't mean to be trite about this, but it can just be simply because the neurotransmitter balance is off. It explains depression, and it explains lots and lots of neuropsychiatric disorders. And so, we need to look at it in a binary way. But what we constantly forget to do is to think about how it is our behavior affects those concentrations of neurotransmitters, and how that can actually change how the system works as well.
So in a way, what we're talking about here, if we're looking at models for human disease, the microenvironment is implicit in having any success in the outcomes. But it goes further than that, because it goes, as you mentioned, that the holistic approach even puts another sort of stratum of intrigue on this.
Yeah.
Let me just take a slight diversion, ask you another question, if I may now. And this has got nothing to do with neuro-oncology. It's just me as an old bloke asking a question, because there was this interesting part of the book where you talked about meditation and comparing the brains of 50-year-old meditators with 25-year-old brains, and stating that the grey matter volume, i.e. the cellular volume, was the same in the 50-year-old meditators as it was in 25-year-old lads and lasses. And so is that anything? Well, two questions are in there. Is this cellular, or is it just a volume thing? In other words, are there more glial cells there in the older brain because they're repairing the damage from the neuronal loss? Or is it neuron for neuron, or is it a mixed bag there? And where have I got to look forward to as somebody who's well past their 50th birthday?
Well, you know what? That's what's really interesting. We have looked at these issues in microcosms, like meditators versus not meditators, but we need to ask these questions of the wider aging society. And by aging, I'm including myself in that. I mean, we're all aging since the day we were born, and we need to understand the effects that our pasts have on our future cognitive abilities, and then look at all the extraneous factors like air pollution and things that actually will affect that too. But to just get back to your original question, mixed bag. I mean, we know that Einstein was very clever, not because he had more neurons, but because he had more glial cells, right? And so he was mopping up neurotoxins, and you know, our brains work on attrition all the time, right? So we lose brain cells all the time, and it's just about how toxic that environment is, and how healthy we are is related to how we manage to keep a handle or a lid on that toxicity. And once that fails, then that's where things start to become more toxic inside of our brains. So part of it is about volume, part of it is about the support systems that are there to keep our brains healthy, but there's another part that has to do with connectivity. And it has to do with how meditation helps us to connect different parts of our brain in a coherent firing pattern. So by doing that, then that means that it keeps the brain healthy, because you're not, it's almost like jump starting a car, and you know, your spark plugs aren't quite right and you're a bit jittery. I think it doesn't happen so much in modern cars, but I'm really old, so you know, it happens. It's a little bit the same in your brain, I think, where it just can get a little bit jittery. There's no oil in the system to keep it going. Cells that fire together wire together. The more cells that fire together and are wired together, the more stable those connections are, and the more robust they are to the ebbs and flows of stress and things that introduce toxicity into the brain, I think. So we need to look at the wider picture as well. Again, we're going back to how our behaviors can affect what's happening inside our heads, and then how that can actually happen things at the smallest scale.
So the situation is in the brain in particular is it's not just about the chiefs, it's about the grease monkeys as well, because if you don't have the maintenance men there, i.e. the oligodendrocytes, the astrocytes, and all the other component cells in the brain, then the whole system ceases to function efficiently.
It just breaks down, and getting a handle on chicken and egg there is really, really important, but also getting a handle on what it is that we can do about that to ensure healthier aging for the masses would be a key to unlocking that particular nut, because we can keep people alive for longer, but we're not doing such a great job on keeping their brains viable for the same amount of time as the body.
They're important points, and again, I go back to the modeling scenarios here, that we need to build in the complete system and not work in isolation. And unfortunately, a mouse isn't a man, and a rat isn't a man, but neither is a cell culture unless we've got all the component parts together, and that includes the appropriate medium in which you're growing things. It includes all the proteins that the cells shoot out. We know that there's not very much extracellular matrix in the brain, but there's an awful lot of things like hyaluronic acid, which we tend to see more about on the television in terms of cosmetics nowadays than anything else. But the brain actually thrives on that, of course. So, I mean, what would you really like to achieve in your career moving forwards? Are there any specific sort of human relevant pathways that you want to explore, and are these things that you can really expect to make major steps forward in the coming years?
I think something that's going to be inherently human relevant and should replace the need for animals. We've got a few things to do on replacing the need for animals in scientific research. One is policy. Obviously, we've got to get away from this validation model that we currently have. We've got to get scientific journals accepting papers that don't have recourse to animals in their validation. So there's two things we need to do there. But I think that there's another thing that actually human research needs to do as well, which is to very much buy into this. I can't just look at a cell without knowing where that cell has come from or what that cell's gone through throughout the course of its life. And I need to know how all the cells work together. And I need to know how all of those cells work together to make you, Geoff, and how Geoff works and what's happened to Geoff in Geoff's life. And so we need to personalize this. We need to have a much more individual approach. Now, this is where data comes in. And of course, we've heard a lot about data good and bad, over the past year or so, maybe 6 months, primarily. But what data will allow us to do in this realm is to sensibly put together data from the cellular work that's happening with a skin sample, a human skin sample on a petri dish, and then move that up through the systems of that person and look at then the behavior of that person and how that then interacts with what's happened to those cells in the petri dish. And that's a huge body of work, and there's a lot of data there, and we're not going to get to that until we start working in an interdisciplinary way. We also have the term multidisciplinary, where you've got lots and lots of different people working on different things, but they don't necessarily interact. And if they interact, then it becomes interdisciplinary. And then we have transdisciplinary, which is a more recent adoption of the word, which is to say, right, okay, we need to go from here to there through these. They all mean pretty much the same thing, but you can see where the nuances lie. But we need to do a lot more of that. We need to remove data from the influence of bias, because data will tell you whatever it is you want based on the question that you ask of it, and it'll give you a yes or no answer. And if your data set isn't full and holistic enough about that person, then it can get flawed. So really we need to work a lot harder on working out what it is, the questions that we need to ask in order to build predictive models for treatments, for example. Would we predict that you, Geoff, would be prone to X, Y, and Z disease? Genetics will tell you something, but your life will tell us a lot more. Your biology will tell us a lot more. We need to put all of those things together. But we need to know not just for you, Geoff, but we need to know for everybody else. And so that we can say, right, okay, we've got this 50-year-old man who has lived his life as such and such, has lived here with these environmental stressors, with a belief in a deity or a dog. And actually that's a really important point, because if you do have an animal, then chances are you exercise, unless it's a cat, because they don't walk very well on leads. But you know what I'm saying? So actually even the kind of animal that people have tells us something. So it's being able to put all of that together in some kind of predictive model so that we can say something about not just the human experience, but the outcomes of that particular human throughout their lifetime. And we work on that kind of thing with trying to predict whether or not aneurysms will rupture or not, because believe it or not, we don't have a measure of that yet. So it's understanding the valence that these factors have. Are they good? Are they bad? Are they positive? Are they negative? But also quantifying the influence that they would have on the model. And that's going to be a never-ending story. But at some point, we're going to hit a tipping point where we can say, you're in pain, Geoff, and you're in pain for this reason. But I think I know what's going to fix you, because I've put all of your data into this model. No. And it says you need to do X. And then that will reduce our reliance on opioids, which have all sorts of negative effects in the brain. And then that will unlock a bunch of positive benefits right there. It's very complicated, but we must not be hypnotized by it.
It is, and it becomes a sort of a super personalized medicine approach, because we all talk about personalized medicine and getting all the genetics together and getting all the ducks lined up and so on. But this really goes one step further, and in fact, in a way, it's going backwards, because it's more like the old GP scenario where the GP knew not just about the diseases that the patient had, but about people probably been there when they were born, and know the family and so on, and would make house calls on a regular basis and so on. This is with an increasing population and a diversity of ethnic origins, and which brings with it other disease prevalences and so on.
I think the trick here, Geoff,
budget's being stretched.
Yeah, and I think one of the things that we seem to be traversing, it's slow, because I'm an interdisciplinary, and I'm director of an interdisciplinary research institute, and so the way we do this is by getting together and sharing and talking and making sure that you check your ego at the door. This isn't about what you know, it's about what you don't know. It's about what it is that we can actually handle together. I don't need to do a PhD in engineering now. That would have been the old model, whereas if I wanted to answer a question that would use in engineering in answering a neuroscientific question, I'd go do another PhD in engineering. I don't need to do that. I talk to an engineer, you know? And it's about making sure that we find a common language, and we actually get together and try to solve these kinds of challenges, these kinds of problems that affect society.
And they're very, very pertinent. I mean, we have MDTs within neuro-oncology, so multidisciplinary teams. Now, way back when I was working at King's College Hospital, King's College London, we started off an MDT. We didn't realise that was what it was then, but we had the neurosurgeons and we had the radiologists, we had the histologists, and we had everybody there, speech therapists and so on, patients coming in. I was there to lead the research team, and I'd go off and talk to patients about what was been on at the last conference I went to in the States and what was changing and so on. And gradually adopting those sort of multidisciplinary teams, I think, has been a great plus to our discipline, whether it's brain tumors or whether it's any other neurological disease or any other somatic disease. They've been handy, but we really need to build on that to a more personal level still. And again, that's going to be a major challenge for years to come. So that's sort of fascinating. I'm aware we're sort of getting towards the end of our allotted time here. There are a whole lot of questions that I've got scribbled down here, which have been sort of gathering dust, and I'm afraid that we're not going to hit all of those. It's been a pleasure talking with you, and I think one of the last things that we probably should address is that, as well as being a scientist and working in a variety of different areas, you're also currently a trustee of Animal Free Research UK. How has that panned out for you? Is that something that you really were wanting to do, and have you learned anything from that experience? And how was that experience for you?
Yes, it's been one of the most rewarding experiences of my life. So I have a long history with Animal Free Research UK. So I was their postdoc at Oxford University way back, and then in my career at Durham University, they funded me to have my own postdoc to further the research of neurostimulation in human relevant science. And then the advert to become a trustee came around, and I thought, wow, I wonder could I give something back? And thankfully they wanted me. I learned so much. I learned so much about how charities work. I learned so much about our wonderful supporters. I learned so much about how it is that we interact with scientists and how important message is, and how it is that we should be communicating what it is that we're trying to achieve by 2040. I learned a lot about governance. I learned so much outside of what I could have learned in my academic career. And actually, it's fed into my academic career, and things that I've done in my academic career have fed into how it is that I've interacted as a trustee. It's been a wonderfully collaborative and reciprocal relationship that I've had as a trustee. So yes, it's been wonderful. It's just been great to be part of this journey to actually give something back. Yes, but actually, I think I've got a hell of a lot more out of it than I gave.
Well, it's been absolutely fascinating, Amanda. Thank you so much for sharing this this facet of your life. I mean, you've gone through the academic work, the neurosciences and so on, and a whole load more as well. So I mean, it's been a very interesting half an hour or more. Thank you to everybody for listening. And if people want to hear more like this, or they want to learn more about animal-free research, the information can be gained by visiting animalfreeresearchuk.org. So thank you for listening, and many thanks, Amanda. Lovely to see you again, and great interview. Thank you.
Thank you, Geoff.